Last night I checked your CV. You're really productive, very productive. You published so many CS papers recently. Wow.
No, no, I'm just you know, have some good people in the lab and
你看到我在了吗？
so is it 9:30 there? 9:30 in the morning?
It's 10:30.
Oh, 10:30. Okay, I see, I see.
Yeah.
So it's 23 hours difference. So how many of these seminars do you have every
It's it's actually it's not a we have no fixed schedule. So
okay.
we send an invitation for to scientists and so it's not a very fixed schedule. So
Hmm. Okay,
case
okay.
by case.
我怎么听？
I think it's time. So can I have a brief introduction?
Yeah, sure.
So okay, so today is a great honor to have Dr. Aska Chu from Harvard Medical School to give us a talk. Talk. So Dr. Chu is a professor of immunology at Harvard Medical School. Um, he started his lab at Harvard Medical School in 2014, and now he is a full
我不
professor.
知道，我也不知道他长什样，对吧？我不知道怎么样。那么我们可以把
Dr. Chu's
开始
research
吗？
focuses
我先把
on
它开，基
uh,
本上这个
sorry, somebody, I need to mute all attendance. So Dr. Chu's research focuses on defined interactions between the nervous system and the immune system, and his lab has discovered many interesting mechanisms by which bacterial passengers active sensory neurons through secrete toxins and proteins to produce pain and itches. So as I mentioned, that Dr. Chu has published a very is very he's very productive, and his lab has published many big papers recently, like cell papers, cell science papers, and Nature papers. And Dr. Chu has been awarded several honors and awards, including NIH Director's New Innovator Award and the Canadian Foundation Innovator Award. And then Dr. Chu has strong connections with China. Actually, I noticed that he's the editorial board of Neuroscience Bulletin. So it's my great honor to have Dr. Chu to give us a talk here. And we have a panelist
Recording in
today.
progress.
And I received a request of recording. So,
Okay.
but I don't know this guy, administrator, who is the administrator. So, uh, Do you mind to record your
No, I
presentation?
don't mind. I don't mind at all.
Oh, okay, thank you very much. So, we have a panelist for today's talk. They are Dr. Yu Fu from Singapore and Dr. Sun Wei from Shanghai Tech.
Hi.
And doc
Hello. Hi.
and
Nice to
Dr.
meet you.
Cao Peng from NIPS and Dr. Li Haohong from Zhejiang University and Dr. Gao Zhihua from Zhejiang University. So please, can you open your camera and say hi to Dr. Chu?
Hello.
Hello.
Hi.
Hello.
Nice
Yeah,
to
good to
see
see
you
you. Nice to
again.
meet.
Yeah.
Good to see you, Fu Yu, and all of you.
I met you in Singapore on the bus. We sit together. We sat
Oh,
together on the
Oh,
bus.
yeah, good to see you again.
Yeah, I work on Microglia. Hi.
Wonderful to see you again. Yes.
Yeah.
so this is my introduction. So Dr. Chu, could you share your slides? And
Yes, I'm trying to share my screen. Can you
oh,
help me?
yeah.
Okay, perfect. Okay. Okay, well, thank you again for your the honor to be here, and thank you for the very nice topic, body-brain axis, which I think is such an exciting area. So today I'm going to tell you about
Yeah.
the role of nociceptor neurons and crosstalk with the immune system in two organs, the gut and the lungs, just to highlight how the nervous system talks to the immune system. So just in terms of the seminar, so I'm going to briefly introduce the topic of sensory neurons and neuroimmune interactions, and then talk about these two organs. So two stories. One, we recently published on how using a chemogenetic screen, we found the role of TRPV1 neurons in regulating Tregs, and then some ongoing work that we're working on, which is the role of nociceptors in regulating the immune system during influenza virus infection in the lungs. Okay, so the sensory nervous system is very important, not just to sense innocuous things in the environment, but also to sense potentially dangerous stimuli. And these harmful stimuli will then induce activation of sensory neurons that drive protective behavioral reflexes. So that includes things like pain or itch or cough. And we're starting to understand that these sensory neurons are coupled to the immune system at the same time as inducing these behavioral reflexes. So it makes sense because these responses are very similar to what the role of the immune system is, which is to protect the body from potential damaging agents, including pathogens and also other damage. So just to very first focus on pain, which is the prototypic nociceptive sensory sensation as defined by actually Charles Sherrington. This sensation we all know, it's the unpleasant sensation that we have experienced, but it's also linked closely with inflammation. So Celsus, the Roman physician from 2,000 years ago, defined inflammation as having four signs: pain, redness, heat, and swelling. So we're starting to appreciate that, every time you have inflammation, there's pain, but also that pain, the nociceptors that mediate pain, signal to the immune system. And this causes a way of protecting us from potentially damaging agents like this heat source here. So nociceptor neurons are sensory neurons that specialize in detecting noxious stimuli. Their cell bodies reside in the DRG, the dorsal root ganglia, which all along the spinal column. They project a peripheral branch to peripheral organs and also peripheral joints and tissues where they sense noxious stimuli. And then they have a central branch which transduces these signals to the spinal cord, where then it is taken up by interneurons and then relayed to the brain to be perceived as pain. I think as an immunologist, what's fascinating is the principles of how these neurons respond to noxious stimuli, and also the fact that they, have so many different ways of sensing different noxious, harmful products. So we know that at the nerve terminals of these nociceptors, there's receptors for noxious heat and noxious cold, for reactive electrophiles, things like ATP and damage-induced DAMPs that are released during tissue damage. And we're now seeing also that they can directly sense microbial products to mediate pain. One of the prototypic ion channels that mediates pain is TRPV1, TRBV1, which was discovered by David Julius as the receptor for capsaicin, which is the active ingredient in chili peppers that causes pain. And he won a Nobel Prize for this discovery because, as we know, it also is a key sensor for noxious heat and also for acid. TRPV1 is expressed in a significant portion of DRG neurons, as well as I'll show you later for vagal neurons, which also innervate the lungs and other tissues. Our lab has shown over the years that neurons can sense microbial products. Some of these are highly damaging pore-forming toxins. So we found that, for example, during infection, these bacteria, Staph aureus and Strep pyogenes, release toxins like streptolysin S, alpha hemolysin, or PSMs, and they form holes in the membrane of the nociceptor. This induces ion influx. Like a cation channel, this will induce firing of action potentials and pain. So this is one of the major sources of pain during infection is actually bacterial toxins acting on nociceptors. We also know that subsets of nociceptors express toll-like receptors like TLR4, TLR5, and these are gated by LPS or flagellin. Rurongji's lab showed that flagellin can bind TLR5. And we recently also helped show that TLR4 signaling can upregulate other ion channels like TRPA1, which then leads to also increased sensitivity to pain. So there are multiple ways by which bacteria interact with nerves, and this is a really fascinating topic. But as a consequence of this activation in the periphery, once the neurons are activated, they also immediately release neuropeptides, which are stored in dense core vesicles. So in the tissues themselves, the coupling of pain to release of neuropeptides allows the pain signal to be coupled to the immune system, because a lot of immune cells actually have receptors for neuropeptides stored in these nociceptors. As an example, substance P and CGRP act on mast cells and macrophages. So mast cells in the tissues are often right next to nerve fibers, and when they detect substance P, they will release inflammatory mediators like cytokines. And so this receptor MRGB2 specifically recognizes substance P. And for macrophages, the calc-RL ramp1 is the receptor for CGRP. And we found, and others have found, that this neuropeptide will polarize macrophages toward the specific phenotype, like an M2-like phenotype. So this allows the pain signal to be directly coupled with the immune system in different ways. This is just a highlighter paper we published last year. Actually, Felipe Pino Ribeiro, who's now a PI at WashU in St. Louis, we were trying to understand how bacteria cause pain when you have infections of the brain. And actually, what we found is that two bacteria, when they invade the meninges, which is densely innervated by nociceptors, Strep pneumoniae and GBS, they activate nociceptors, and the nociceptors then release CGRP, and this again polarizes macrophages into a kind of actually anti-inflammatory phenotype. This helps the bacteria because then the bacteria now can get through this barrier because the macrophage response is more shut down. So this pain response actually is hijacked by bacteria to help them invade the brain. So this is just really interesting dynamics between microbes and pain fibers. So there's a lot of really exciting work in recent years looking at the role of nociceptors in different tissues and how they modulate the immune system. So often a lot of these work happen in barrier sites like the skin. This is the meninges around the brain and the gut, because these barrier sites have a lot of nociceptors. They talk to innate and adaptive immune cells in these tissues. But also very interesting recent work has also shown that nociceptors in the spleen can regulate B cell responses. So this is a really interesting area. Like, how do all the different immune cell types recognize signals from pain fibers, and how does this change the immune response in different settings? So now I'm going to focus first specifically on the GI tract. So the GI tract is one of the areas where we think there's a lot of crosstalk between neurons and immune cells. And we also know that when you have infection or colitis or celiac disease, these kind of inflammatory diseases of the gut often has pain. So there's a lot of interest in defining how visceral pain occurs in these disorders and the gut-brain axis. So is there a role for the microbiome or immune cells in the gut to cause pain? And also, how do the nociceptors that innervate the gut talk to the immune system or other cell types in the gut? So recent work actually from Daping Yang, who's here actually on the Zoom, I just saw him, really nicely showed one of these aspects of crosstalk. So this was work published a couple of years ago in Cell, where Daping and Amanda, another postdoc, showed that nociceptors, when they recognize things like capsaicin or also microbiome-derived products, will release CGRP, and these talk to mucus-producing cells called goblet cells in the gut. So the nociceptors will release CGRP, then goblet cells will produce mucus. And mucus is very important to line the gut and actually serve as a barrier against the potentially harmful microbes, but also for homeostasis of the gut, for the microbiome, and to protect against things like colitis. So this is actually a nice image that Daping took of goblet cells in red here, and the nerve fibers right underneath the goblet cells that are talking to these mucus-producing cells. A very interesting role of nociceptors in homeostasis of mucus in the gut. Another lab, David Artis' lab, also showed a role for nociceptors in microbiome homeostasis. So we wanted to kind of take a broader approach of finding novel gut neuroimmune interactions. And this is work that we did with Christophe Benoit's lab. So Christophe is one of my colleagues at Harvard Medical School who's a leader in T-cell biology. And so Kim in my lab, who's a postdoc, she's now at the Mayo Clinic, and Yang Zhu, who's a postdoc in Christophe and Diane Mathis's lab, teamed up. So we wanted to find approaches to define novel neuroimmune interactions. And we also collaborate with Steve Liberles and also other members in the lab, including Daping, to look at this. So just the question we had is, the gut is so intricate in terms of the nervous and the immune system. So this is just to show you different segments of the gut are innervated extrinsically by both sensory neurons that I talked about earlier, like nociceptors from the spinal cord, but also you have sympathetic neurons and also vagal neurons that innervate the brainstem. This is just another view of this, that there are multiple subtypes of neurons that innervate the gut from the outside. But also there's the enteric nervous system, which resides fully within the gut, including cholinergic neurons in the muscularis and submucosal plexus. And all of these neurons mediate peristalsis and motility, and also pain and other functions. This is to also show you that, within each of the systems, there's also a lot of molecular heterogeneity. So we want to find a way where we can target distinct molecular subtypes of neurons in the gut, and also both extrinsic and intrinsic neurons, and then see the consequences on the immune system. So these are actually a lot of the markers we use. You can see some of them are shared between different types of nodos or DRG neurons or enteric neurons. And the approach we took is chemogenetics. So many of you may know this approach as neurobiologists, where we can use engineered receptors to drive activation of neurons. So here what we're doing is we're taking a viral AAV virus called PHPS that targets peripheral nervous system. So Viviana Gradinaro has developed this. And then we expressed an activating dread virus. And this we use in a Cre-dependent fashion. So this virus is dependent on the presence of Cre, and we use 8 different Cre lines. And what we did is we infected neonatal mice. So this allows more efficient labeling of these Cre subtypes. It's not going to mark every single Cre positive neuron, but a significant proportion of them. And then what we did is we waited until the mice got to adulthood and activated these neurons using the synthetic ligand CNO. And what we did is we activated them over the course of about 2 weeks. So every other day, followed by detailed immune profiling by flow cytometry. So taking advantage of Christoph being an expert on the gut immune system, we had a panel of about 24 markers where we can look in an unbiased way at all the different immune cell types and how they change after activating different neurons. So this is kind of the matrix of different neurons that we targeted. We use 8 different Cre lines, TRPV1, piezo2, MRGD, and NOS VIP. So a lot of known markers for different gut innervating neurons. And here we're using the AAV DREAD, which has an M-Cherry reporter, and we can confirm that it labels subtypes as we might expect. For example, TRPV1 neurons mainly are in the nodose ganglia and DRG, whereas something like CHAT is not really in these extrinsic sensory fibers, but more in the myenteric plexus of the ENS. So having confirmed that a lot of these viruses are getting to the types of neurons we expect, then we did the chemogenetics and did the flow analysis. And this is kind of a summary of a lot of the data that we found. So what you see here is on the axis here are the subtypes of neurons that we activated, and 3 different segments of the gut that we then looked at by flow cytometry. And then on this, you can see on the x-axis, these are all the different types of immune cells that we can detect by flow cytometry. And we're only highlighting changes that occurred that were significant. So what you could see is that some neuronal subtypes caused, actually a lot of them caused suppression of different immune cell types. So just to highlight some things like NOS1 neurons will cause suppression of Th17 cells, specifically in the ileum, but CHAT positive neurons suppressed neutrophils, specifically also in the ileum. Whereas the one that we thought had the biggest changes is actually TRPV1 neurons, which are nociceptors. This one you can see had a lot of effects on multiple populations of cells, but mostly in the cecum and the colon. And in particular, we were very interested in the effects it has on RA gamma positive, FOXP3 positive Tregs. So I'm going to talk a little bit about that. So T regulatory cells, many of you may know, are critical modulatory T cells. So the peripheral or induced Tregs are important in the gut, in particular, to suppress aberrant immune responses against the food that we eat and also against the microbiome. So these are all foreign, and they're foreign antigens coming from these sources. So you need to have regulatory T cells to prevent inflammation against these. So there are 2 flavors of of Tregs in the gut. There's Helios positive and RO gamma positive Tregs. And what we found was that if we activated the TRPV1 positive neurons chemogenetically, you could see this significant downregulation of this RO gamma positive subset of Tregs. And here is looking at the colon. You see there's no effect by the other subtypes. So this is somewhat specific. So we also looked across other organs. For example, in the lung, we didn't see this effect. Although we did see, we also did not see it in the mesenteric lymph node, but we did see also some downregulation in the spleen. So this, anyways, TRPV1 neuron activation leads to downregulation of these RR gamma Tregs. So what is happening here? Well, one of the things that we found that could be happening is related to proliferation. So if we looked, now looking at B cells and CD4 T cons or CD8 T cells, but looking at Ki67 as a marker of proliferation, we did not see effects of TRPV1 neuron activation on their proliferation. But for Treg subsets, and here actually total Tregs as well as RR gamma positive and Helios positive Tregs, you see that after neuronal activation, there's less Ki67 expression in the cecum and the colon. So that could be one mechanism. We also found by single-cell RNA-seq after neuronal activation that there is a lot of transcriptional changes occurring in the immune system. And actually not just in the Tregs, but also in myeloid cells. And some of this included the upregulation of cell stress-related signatures and downregulation of myeloid activation and migration signals. So the neuronal activation is inducing cellular changes in the gut. That includes transcriptional changes. So you might expect if you have downregulation of Tregs, you might get enhanced inflammation if you challenge the gut. So that's what we see here. What we did is we did the chemogenetic activation of the TRPV1 neurons, followed by DSS colitis, which damages the barrier of the gut. And you could see that the TRPV1 activated mice lost weight more than the controls. And you also see increased histological damage by H&E staining. So as neuroscientists, one question is, what are the neurons that mediate this? So I mentioned that TRPV1 is expressed in both vagal and DRG neurons. So the first thing we did was try to kind of target more specifically each of these subsets. So here what we did is took the AAV reporter dread virus and injected it into TRPV1 Cre mice, but only into the vagal ganglia, the nodose ganglia. So here you could see this approach labeled just nodose neurons, but not DRG neurons. And then when did chemogenetic activation, this had no effect now on the Tregs. Whereas if we did an intrathecal injection of the virus, so this will now kind of drain to all of the dorsal ganglia along the spine, you could see it labels very nicely DRG TRPV1 Cre positive neurons, but not nodose ganglia neurons. Here now, when we did chemogenetic activation of neurons, it phenocopied the whole TRPV1 Cre body activation. So you said downregulation of colonic and cecal RR gamma Tregs. So we think it's DRG neurons that mediate this phenotype. What is the molecular mechanism of the neuronal suppression of Tregs? So I mentioned there are a few neuropeptides that these neurons produce when they're activated and release into tissues. So the two candidates we looked at is CGRP, which is encoded here by alpha or calc-A, which is alpha CGRP, and also TAC1, which encodes substance P and a neurokinins. So what we did is we crossed the calc-A knockout or TAC1 knockout onto the TRPV1 Cre background, infected with the AAV dread virus, and repeated this activation. And you could see that knocking out CGRP, now these mice no longer show downregulation of RR gamma Tregs in the cecum of the colon, whereas there was no effect of knocking out substance P, which is encoded by TAC1. So CGRP is one of these neuropeptides which signals to many types of target cells, including immune cells, via its co-receptor complex formed by RAMP1 and calc-RL. So RAMP1, calc-RL is a G protein coupled receptor, which upon recognition of CGRP signals via adenylate cyclase. So what we noticed was that if we look at single-cell RNA-seq data of gut Tregs, the RR gamma 1 Tregs actually have good expression of this receptor RAMP1. So this led us to hypothesis maybe the neurons are actually talking directly to these Tregs and causing their transcriptional changes and downregulation. So if we stained mucosal gut tissue for CGRP nerves, you can see that kind of network here. We see them often also close to FOXP3 positive Tregs. So if we now did this larger cross where we crossed RAMP1 flocks, where we can remove the CGRP receptor just from well, FOXP3 positive cells, which includes Tregs. And then we crossed them again with the TRPV1 Cree and then did the chemogenetic activation. This now ablated the effects of chemogenic neuron activation induced Treg suppression. So we think that at least part of the signal from is through CGRP acting directly on Tregs. So for the kind of summary of this first part is more big picture is that I think there are a lot of complicated interactions between neurons and immune cells, and we're just looking at the tip of the iceberg now. We use chemogenetics to try to reveal novel interactions where we activated neurons, and this led to discrete changes of different immune cells in the gut. In particular, we found that TRPV1 neurons, which are nociceptors, suppress Tregs. And this is in DRG, not NODOS, and this is through CGRP ramp 1 signaling. What's kind of interesting is that in the pain field, a lot of people are trying to pursue actually using Tregs to block pain. So if pain fibers suppress Tregs, there's an interesting counterbalance of these two signals, I think, the regulation of immunity versus inflammation and pain. So now I'm going to switch gears to talk about another body-brain axis through nociceptors, which is the airways, the lungs. So another key mucosal organ, and I think, again, the neurons play a key role in regulating the immune system in this organ. So the lungs are also innervated by nociceptors, so they may not necessarily mediate pain per se, but many of them mediate cough and other airway protective reflexes. This is actually some beautiful images of the trachea being innervated by TRPV1 positive nociceptors that also express CGRP. This is now deeper into the lungs and near the alveoli. Very nice work from Steve Liberlese's lab, who's a close collaborator, has shown that there are many subtypes of sensory neurons that innervate the lung, including TRPV1 neurons, but also many molecular subtypes. So he's shown that there's probably dozens of vagal neurons and several of them innervate the airways. This is a very nice Cell paper that he published a few years ago showing that if he optogenetically activated one subset that expressed P2RY1, this will actually cause like stoppage of breathing, so apnea, whereas activating this subtype, MPY2R, actually induces increase or hyperventilation. So these neuron activations will induce different reflex circuits. We want to know the role of neurons in immune response. So the specific question we asked is, what about in the setting of influenza virus infection? So actually, I don't have a slide here, but I should say that this work was done by multiple very talented people in my lab, including in particular Daping Yang again, who I mentioned did the very nice elegant mucus work, as well as Nicole Almanzar, who is a graduate student in my lab, and Jingya Xia, who's also a former postdoc, and Swalpa Udit. So these are drivers of this project, as well as Pankaj Baral. So many, many people in my lab.
Recording stopped.
Sorry. So we wanted to know what happens if we target TRPV1 neurons in flu infection. So what we did here is we use a chemical ablation strategy called RTX, which will kill off TRPV1 neurons versus vehicle control. And we gave them then intranasal flu infection, PR8. And what you can see here is if you get rid of the TRPV1 neurons with RTX treatment, you get significantly worse survival outcome, where the mice die more. Actually, there seems to potentially also be a sex difference because female mice die more than even more. So this worsens survival. But interestingly, if you look at actually recovery of viral transcripts, so here looking at nucleoprotein levels at each of these time points post-infection, there's no difference in the viral transcript. So we don't think it's because the virus is being cleared less in the RTX mice. Also, we see that the vagal neurons in these mice are responding in some ways. So this ATF3 is a marker of nerve injury. And actually, at different time points post-infection, ATF3 is going up in the vagal ganglia. So the vagal ganglia I mentioned are the key source of innervation of the lungs. So here we targeted those vagal neurons using DT approach to kill off TRPV1 vagal neurons. And this phenocopied the total TRPV1 neuron ablation RTX approach, where they also did a lot worse in after infection compared to PBS control. So what is happening if there's not a difference in viral load? Could it be inflammation, right? So we know that influenza causes a large amount of inflammation and a cytokine storm, which can cause pathology. So actually, if we look in these RTX treated mice, this other way of targeting these nociceptors, and we look in the lung, especially at the later time points, they have a lot more fluid accumulation and immunopathology compared to the vehicle control. So this was also accompanied by like cytokines going up, going a lot more as well. In the absence of these nociceptors, if you look at this is a 44-plex cytokine array, many different cytokines actually went up more so in the RTX treated mice compared to the control treated mice. And this is just, to highlight a few examples, like things like IL-6 or CXCL1 or IL-13 are going higher in the nociceptor deficient mice compared to control. So there's uncontrolled inflammation. So this is with cytokines. What about the immune system? So what Nicole and Jingya and others did here was to do single-cell RNA-seq of immune cells from these either naive or infected mice at day 7, which is the peak inflammation time point. And this is just to show you there's a UMAP, of course, of many different types of immune cells that accumulate during infection and also at homeostasis in the lung. And also we could detect viral transcripts themselves, and a lot of the flu virus is actually accumulating in these macrophages and monocytes during infection. So what about if you compare now between this vehicle versus RTX, so the ones lacking neurons in RTX mice versus vehicle control? The RTX mice had a lot more certain myeloid cell populations like neutrophils and interstitial macrophages, whereas they had decreased numbers potentially of B and T cells. So this is interesting. So there could be a shift in the myeloid population in particular. And actually, if we look, for example, in the BAL fluid, which is the bronchial lavage fluid, there is indeed a lot more immune cells generally, but in particular neutrophils and monocytes, which are the pro-inflammatory myeloid cells in the airways in the absence of these nociceptors. So these could be driving the damage to the lungs and potential death to the mice as well. In addition, looking by the single-cell RNA-seq, we also looked at like cytokine response signatures. And so you know that with viral infection, you need a type 1 interferon response to protect against the virus. But that also showed a decreased signature in the macrophages when you have an absence of nociceptors. And also Nicole, who is a single computational biologist as well, looked more detail at the single cell data, and there's specific subsets of neutrophils that show up only in the viral infected lungs. And this particular one is increased in the RTX treated mice over vehicle. So the neutrophils can be very damaging in the airways when you have infection. So just to show you a final piece of data, we thought, okay, maybe these myeloid cells or these neutrophils in particular are damaging the airways, and you have too many of them in the absence of neurons. So we treated the mice with anti-GR1, which will kill off the neutrophils. And so you can see that here. And what you could see here is the RTX treated mice treated with vehicle isotype control versus the vehicle treated mice with isotype control. So the absence of nociceptors causes worse survival, but if you get rid of these neutrophils and monocytes, you can restore some of that survival. So for this second part, final part, I think I hope to show you that in the lungs, the neurons actually mediate airway protection, and part of the mechanism is actually by dampening harmful inflammation. So the neurons control pro-inflammatory cytokine production and also neutrophil recruitment. And if we get rid of these downstream harmful immune responses, you could start to rescue the mice from inflammation and damage. So it just shows, I think, the neurons are very important to control immune responses in the gut and also in the lungs in this case. And targeting neurons may be a novel approach to treat respiratory infections. So I mentioned a lot of the really great people who did the work in these projects. Actually, I think this slide is not. So I should have highlighted different people. Actually, this probably for a different talk. But anyways, Da Ping I mentioned, and Nicole did a lot of the flu work. And then I mentioned earlier work on the gut was actually done by Christophe Benoit, Yang Zhu, and also Kim Mearshart. So thank you for your time. Happy to take questions.
Thank you very much. Yes, we very great talk. And okay, if you have any questions, please raise your hand in the chat box, or you can type your questions directly. The first question is from Liu Chenxin. Liu Chenxin, are you there? Could you answer your questions by yourself, or do you need me to read your questions? From Zhejiang University, Liu Chenxi is a graduate student.
Yeah, I see the
Yeah.
chat. Are you
Yeah,
there?
I can.
Okay.
Yeah, hi Isaac.
Hello.
Thank you for your wonderful talk. And I'm a first year graduate student at Zhejiang University. Here are my two questions. The first one is, are regulatory the Tregs the only immune cells suppressed by TRPV1 neurons? And do other cell types such as Th17 cells or ILC3 also
Yeah,
respond
that's a
to
very good
those
question. So
neurons?
I only briefly showed the heat map. Actually, there are quite a few other cells also suppressed by TRPV1 neurons, including particularly macrophages and ILC2s, I believe. So there was not much change to Th17 cells, but ILC2s and macrophages were also suppressed. What was kind of interesting is when we targeted CGRP, we only see the Treg effects actually go back restored, whereas we don't see much different. So there's probably some other mechanism that the neurons are talking to macrophages. Does that answer your question?
Yeah, yes, that's really exciting because we all know that the Th17 and the Tregs are the balance of the
Right.
immunity. Yeah, and the second question is, are there any upstream neuronal circuits like some specific brain regions that modulate immune cells through these
Yeah,
TRPV1 neurons?
that's an excellent question because if there's top-down control, and I think that's one of the big questions in the field is, are there possible, regions of the brain that respond to peripheral inflammation, and then they feed back down through the peripheral nervous system to target those immune cells. So the short answer is I don't know. I think that could be very interesting. Right now, we're starting to map brain regions that respond to different